The stories of dinosaurs' lives may be written in fossilized pigments, but scientists are still wrangling over how to read them. In September, paleontologists deduced a dinosaur habitat from remittents of melanosomes, pigments structured in the skin. Cetosaurus, a spectacled dinosaur about the size, had a camouflaging pattern that may have helped it hide in the forest. Jacob Winther and colleagues say the dinosaur was very much on the bottom of the food chain, says Winther of the University of Bristol in England. It needed to be inconspicuous. Identifying ancient pigments can open up a wild new world of dinosaur biology and answer all sorts of lifestyle questions, says zoologist Hannah Rowland of University of Cambridge. You might be able to take a fossil and infer a dinosaur's life history just from its pigment pattern, she says. That the most exciting thing, not so far, says paleontologist Mary of North Carolina State University. Evidence of ancient pigments can be ambiguous. In some cases, microscopic structures that can appear to be melanosomes may actually be microbes, she says. Both hypotheses remain variable until one shot down with the data. Until then, she says, inferring dinosaurs' lifestyle from alleged ancient pigments is impossible. Author's work, published in September 26, is the latest in all simmering debates in the world of paleocolors. The study of fossil pigments and what they can reveal about the ancient animals. Disputes over his team finding and what needed to clearly identify fossilized melanosomes point to current pitfalls of the field. But the promises is clear. Paleo is color could paint a vivid picture of dinosaurs' life, offering clues about behavior, habitat, and evolution. This is a crucial new piece in the puzzle of how the past looked, Winther says. Cetosaurus was a parrot-beaked herbivore about the size of a large dog. Researchers found signs of pigmentations on its tail region, back leg, and elsewhere that hint at its habitat. Scientists have been puzzling over animals for centuries, but 8 years ago, paleontologists got a wake-up call. That's when Winther and the colleagues proposed that microscopic structures in roughly 125 million years old fossil feathers were actually a type of melanosomes. These pigments pouches rest inside pigment cells and in this particular fossil feather might have delivered a blackish hue like a black bird. Scientists have noticed similar structures inside fossilized skin and feathers since the early 1980s, but people assumed that these structures were remnants of bacteria, perhaps decomposers that feasted on the dead animals, says paleontologist Martin Sanders of University of Bonn in Germany. The new colorful interpretation sparked a flurry of research, and scientists have been spotted what appears to be melanosomes of all kinds of fossilized animals. Paleontologists, in fact, is now awash in colors and patterns. Pigment pods may have been painted reddish-brown speckles on the face of the late dinosaur theropod, brushed chestnut stripes on a long-tailed dino from China, and made the plumage of four-winged dino called Microraptor iridans. Primary dinosaur probably had a weak, glossy iridescence of all over its body, says evolutionary biologist Matthew Shockey of Ghent University in Belgium. His team deduced Microraptor's colors from the shape of its melanosomes. Modern melanosomes generally carry a mixture of two melanin pigments, dark brown-black elenium and red-yellow phenomenon. Scientists have linked colors in mammals and birds to melanosome shape, a meatball shape for reddish-brown hue, for example, and a sausage shape for dark colors. The iridescent feathers melanosome tend to be even thinner, Shockey says. Microraptor's melanosomes looked like skinny sausage similar to those seen in the feathers of modern crows and ravens, says Shawkin, who reported the finding with Winters and colleagues in Science 2012. 3 years later, Winther laid out the case for inferring colors and ancient history from fossilized pigments in a review in Bioessays. Not only can the distinct shape of melanosomes offer clue, he noted, but chemical tests can help to detect the presence of melanin itself. Finding the pigments in fossil, he argued, put the old bacteria hypothesis to rest. Mary and colleagues disagree with Winther's take in the review published in Bioessays later in 2015. Researchers need to be cautious when deducing the hue of extinct animals, the scientist wrote. Any melanosomes looks alike in a fossilized feather or skin could actually be microbes. After all, microbes are everywhere. These animals die in an environment that was not sterilized and free from microbes. Mary says, think about it. If you take a piece of chicken and throw it out of your window backyard, how long does it take for a microbe to overgrow that chicken? Tiny organisms are hardly two, but both microbes are sticky biofilm. They form preserved in the fossil record, and Mary says microbes and melanosomes overlap completely in a shape and size, which make the two tough to tell apart. What's more, some microbes actually make melanin themselves, detecting the pigment in the fossil and is not rock solid sign that the Asian animal was black, brown or freckled. It's not that. Mary or bioessay co-author Johan Linder-Green, a geologist at Lund University in Sweden, doubted that the melanosomes can leave a trace in the fossil record. The issue, Linder-Green says, it's that not all round structures you find are melanosomes. Chemical tests could help distinguish the two bacteria, for example, leave behind the traces that can be identified with pyrolysis, gas chromatography, mass spectrometry, but that requires sample to be vaporized. It can destroy much of what you're trying to study, says geologist Roy Wilgis of the University of Manchester in England. So it's not always possible. Winther's new work isn't likely to settle the debate. In fact, people were arguing both sides in October in a meeting of the Society of Vertebrate Pathologists in Salt Lake City. Arena Roy, a Bristol colleague of Winther's, reports size difference between fossilized melanosomes and bacteria growing in a decaying chicken feather in a lab. Alinsons Moore, an NC State colleague of Mary, said that looking weren't enough. Finding keratin, a protein that typically surrounds melanosomes, could serve as evidence for pigments in fossil. The fossil described in Winther's new paper is spectacular. Mary says, it's got skin all over the place. I can't think it's too many dinosaur specimens that are preserved like this. The dinosaurs lie on his back, flattened on the slab of volcanic rock, skin covered in a complete intact skeleton and dozens of long bristled spokes from the tail. Cetosaurus is a herbivore that lived for some 120 million years, walked on two legs, and could have reached half the size of the height. It would have been a super cute animal. Winther says it's got the wild face and a look a bit like E.T. Black material speckles the dinosaur's body, tail, and face. Winther believes that the material is an ancient remain of a pigment. His team examined sampled chips from the fossil and saw what he considered the telltale robes of melanosomes. Mostly impressions in the rock, but also some microbodies, the 3D structure themselves. Based on the dinosaur's pigmentation pattern, it would have a dark black that fades to a lighter belly. The type of coloring called countershading shows up in the animal from penguin to fish and may act as a form of camouflage. It lightens part of the body typically in a shadow and darkens part typically exposed to light. If you want to hide, it makes sense to try and obligate the shadows, Roland says. The prediction to diffuse light matched the model painted in Cetosaurus. It's like what we see live in the forest, living animals. Winther says this thing was a camouflage. Going from fossil to forest may be more of a leap than a step, other scientists suggested. Cetosaurus skin very well may contain ancient pigments. Wallace says, I don't think it's a crazy idea, but he adds to Winther's group, I don't think they have proved what they claim. Winther's team, for example, used 4 tiny fossil samples to explorate the coloring of a whole dinosaur. I think it's a bit overreach, Wallace says. Mary also noted that the specimen was varnished, presumably to protect the bones and the soft tissues. It helped and it happened before Winters and the colleagues got their hand on the dinosaurs and make it possible to perform a chemical test that would bolster the claim of pigment. Varnish is a horrible destructive fossil, she says. It totally ruins the specimen for other type of analysis. Winther argues that his team has chemically analyzed other fossils and found the evidence of melanin, not bacteria. The microbodies in those fossils just look like one of the Cetosaurus, he says. Winther's team also saw the evidence that one of the kind of microbody and his distinct round shape. If the structures were actually bacteria, he says, you would expect to see a whole range of shapes and size. Some of them would be shaped like corkscrew, some would have been fragile, some would have been homogeneous, some would have been tiny. That's the tricky part of the bacteria, counters Lindgren. In some cases you have a huge contraserum, but in other cases you have one single type. Sanders' interpretation has its supporters. It was a spectacle at first, Sanders says, but now there has been such an array of these little bodies, it's such pretty clear that at least some of them are not bacteria. Despite some continuing controversies, Sanders says many paleontologists now accept the microstructure in fossil may be melanosomes. Addition to researchers through would help the entire community, he says, so there is no longer any lingering doubt. Along with the chemical test, Mary suggested researchers could also transmission electronic microscopes, a technique that blasts an electronic beam through a tiny sliced sample. With the TEM, melanosomes appears as black blobs. Bacteria tend to look different, in some cases more like fried eggs. Shockey, for one, is looking to the chemistry in a paper published in Online November 14 in a paleontologist. His team used a technique called Raman spectroscopy to build a case for feather colors in a bird that died some 120 million years ago. In the feathers, the researchers spotted the skinny sausage of iridescence of melanosomes and chemical signs of the pigmentation elysium. Shockey thinks the chemical evidence could help head off the criticism that might encounter. Working through the field snags, paleontologists might come together to fill the hues and tints and potentially the habits and habitats of the ancient animal that until recently has been known primarily by their bones.